In this video, we are going to learn about the blood groups and blood transfusion. So, basically, if I tell you about the blood groups, here we will discuss about ABO blood groupings. Blood transfusion means transferring the blood from one person to another. So, this ABO blood group system was given by a scientist Carl Landsteiner in 1900. So, ABO blood grouping is based on the antigens. Antigens are basically the glycoproteins, and these antigens are present on the surface of RBC. So, therefore, it is written here that Carl Landsteiner discovered that human beings have glycoproteins on the surface of RBCs called antigens, and another type of protein, gamma globulin, in the blood plasma called antibodies. From here, you can understand that antigens are found on the surface of RBC and antibodies that are represented by AB are found in the plasma. See, here in this diagram, ABO system is shown. That circle, the outer one circle, this one represents your RBC, and the rim of the circle is the RBC surface. So basically, on this RBC, antigen A is present. Fine. So the blood group is A. Means you have to name the group of the blood based on the presence of antigen. Similarly, in the blood group B, you will have antigen B on the RBC surface. In blood group AB, see, it is AB means it will have A as well as B. Therefore, antigen A also present and antigen B also present. Blood group O. This O resembles 0 means no antigens. Therefore, there is not the presence of antigen. Now comes the ABO blood groups. So basically we know that two types of antigens are found, A and B, on the human RBC. And based on the presence or absence of these antigens, as we have discussed in this diagram, the blood groups falls into four blood groups, that is AB and O. As we have discussed here also, four blood groups. This forms ABO system of blood groups. Now comes the antigen and antibody interaction. To understand about antigen and antibody interaction, first of all we have to understand the presence of antigen and antibody. As I have told you that the name of blood groups come from the antigen presence. So A blood group means A antigen, B blood group means B antigen, AB means both, and O resembles 0, therefore none. Now antibody means the body which is against the foreign body. If I tell you that antigen on RBC is A, so this antigen is A. Now the foreign thing will be for this blood group will be B. So for the production of B, we have something that will attack this B. Therefore, that molecule, that gamma globulin, is named as anti-B. Similarly, for the blood group B, A antigen is external disturbance. Therefore, antibody found is A. In the blood group AB, the antigen A and B both are present. There is no foreign body. Therefore, none is written here in antibody section. Similarly, in blood group O, that resembles 0, it doesn't have any kind of antigen on RBC. Therefore, every kind of body is external for this O blood group. Therefore, antibodies found in plasma are anti-A and anti-B. Now, what happens? Two types of antibodies are found, that is anti-A and anti-B antibody. Person with one antigen have natural antibodies in their plasma against other antigen, as we have seen also in this table. Now comes the clumping reaction. Basically, clumping reaction is a reaction that happens in the blood between antigen and antibody. What happens? Suppose this is blood group A. Fine. So, on the surface, it will be having the antigen A and the antibody B will be present in the plasma. This is the scenario. Now, what happens? This patient, or you can say this person, needs some extra blood, and the donor of the blood from whom you are taking the blood and transferring to this body, suppose has the blood group B. So, what happens? This blood group B person will be having antigen B on the surface and antibody A in the plasma. So, what will happen? They are incompatible because antigen A will react with this antibody A and antibody B will react with this antigen B. So, what will happen? There will be a clumping, or you can say a lump formation. Fine, and destroyed RBC in the blood group. That is known as clumping reaction. Therefore, here it is written that antigen A of blood group A and anti-A antibody from blood group B are not matching. So, such kind of conditions are known as incompatible. Similarly, antigen B and anti-B antibody are also incompatible. It means when blood from blood groups A and B is mixed, clumping or lump formation occurs due to antigen antibody interaction, or simply you can say antigen antibody reaction. I hope the topic is clear to you. Thank you.